Okay, so I just like to cover the structure and detail of one of the three structural categories of neurons. These are called multipolar neurons, and they're the most abundant types of neurons that we have. They're also the ones that you learned way back when we did histology. So as far as the parts of the neuron, there's also a little table here that goes through the parts right there. But let's go back and look here. Okay, so parts of the neuron. This portion right here that has most of the organelles in it is called the cell body, or sometimes the soma. It contains the nucleus, most of the other organelles. It tends to have this dark staining because it's got lots of rough ER. That makes it kind of look grainy. They call them chromatophilic substances. It's basically the rough ER. Most neurotransmitters are amino acid based. So there's a lot of that that's going on inside the cell body. Okay, the nucleus, remember from way back when, usually has kind of light around the outside and then dark in the middle. So the nucleolus is usually pretty dark. So it's called an owl eye nucleus. It's not always super easy to see, but the nucleolus in the middle is darker than the whole nucleus. Okay, so where are these? Generally speaking, the neuron cell bodies are mostly in the CNS, not very many of them in the PNS. When they're in the PNS, they're in little blobs. I showed you all the way back at the beginning. You can see one here too. When a neuron cell body exists in the peripheral nervous system, it's usually protected in a little blob with some of its buddies, and this is called a ganglion. But when you have a big collection of neuron cell bodies in the CNS, a lot of times it's called a nucleus. I know that's like strange double use of the same word, but we will run into some cerebral nuclei, some hypothalamic nuclei, some thalamic nuclei. It basically means there's a blob of neuron cell bodies in this location and somebody figured out what they did. Okay, so processes attached to neurons. We did this back at the beginning as well, but we're going to look at it in more detail. So the processes that are attached to the neuron, that there's more than one on a multipolar neuron, and it's gathering information and bringing it toward this neuron cell body. Those are called dendrites. In a multipolar neuron, there's going to be more than one dendrite. So here's a bunch of dendrites that you're seeing right here. So those are all dendrites. They carry information toward the cell body. They're relatively shorter than an axon. And for those of you who have had phys, they can generate local or graded membrane potentials. They have lots of receptors on their surface. So that's part of the reason that information is going toward the cell body, because they can receive information from other things. And then axons, there is only ever one axon, okay? It can branch as many times as it wants once it leaves the neuron, but it only has one attachment to the neuron. This portion right here, what attaches to the neuron, ends up being physiologically important. It's called the axon hillock. And what this will do is, this is the portion of the neuron that's able to generate a really fast electrical stimulus called an action potential. So the axon hillock is the first portion. For those of you who have had phys, this is the first place where we find the voltage-gated ion channels. For those of you who haven't, don't worry about it. Axons can be super long. For instance, the axon that controls my little toe, the cell body is up in the lumbar region of my spinal cord. Okay. When axons are super duper long, sometimes they refer to them as not nerves, but nerve fibers, which is one of the little parts. If I teased apart a nerve, what I would see. Okay, now axons can branch in the middle, and these branches can continue to form. This is called an axon collateral. It's still controlled by the same axon, and it's going to work with the same axon, but it's branch in the middle. So they can branch in the middle, and then they do branch at the end. At the very end are these branches called telodendria, and telo means end and dendro means fingers. So it's literally the fingers at the end, the telodendria. And then at the very end of each of the fingers are what looks like little ET fingertips. Those are called, sorry, no one ever got together on what to call them. either synaptic knobs, which they're called here, okay, or axon terminals or end bulbs or terminal boutons. Sorry, nobody got it together. You can use what's in your textbook. They don't use the same thing in the phys textbook, but why are they shaped like that? That's actually where you store the neurotransmitters so that you can release them and, for instance, communicate with another cell. And then some, but not all axons have this coating around the outside, which is called the myelin sheath. I'll tell you how that's built in just a minute. The job of the myelin sheath is to insulate the axon and make sure the action potential, the electrical signal, goes really, really quickly. If you take the myelin off, this thing will not be able to send the signal either at all or quickly enough to do any good. Now, one of the other things is that this myelin right here that's wrapping around the axon tends to make the tissue white. So wherever you have a whole bunch of myelinated axons, the tissue tends to look white. So white matter tends to be myelinated axons. So let's see. They are, since myelin is basically, let me show you how it's actually made. So when you take an axon and you myelinate it, there's a couple of ways that you can do it. Let me show you one way to do it. So notice that's what's wrapping around the axon. Kind of looks like a sausage lengths here. But if you look at it closely, what's actually wrapping around the axon is one cell is taking its entire self and wrapping and wrapping and wrapping its cell membrane around the axon. So myelin is mostly made of phospholipids because cell membranes are mostly made of phospholipids. So if I took this and I did a really, really cool microscopy on it, here's the axon and here's this layer after layer after layer of myelin wrapping around it. So how does that actually occur? Well, if you are in the peripheral nervous system, this is the way myelination occurs. So if that axon is running through the peripheral nervous system, there's a cell, it's one of the glial cells, and it's called a neurolemocyte or sometimes a Schwann cell. And this one just wraps its whole little body around and around and around until you get this thing really well insulated. And that is the myelin sheath. And like I said, it's mostly made of cell membrane, which is mostly made of phospholipids. So this is how it's done in the PNS, and it ends up looking like this, like we just looked at. But how it's done in the CNS is with a different kind of cell that kind of does something similar. This cell sits off to the side, and it wraps fingers of its cell membrane around different axons. So this is how you myelinate in the central nervous system. The cells that do it are called oligodendrocytes. Oligo means many, dendro means fingers, and cyte means cell. So it's a many-fingered cell because it's wrapping like itself around a whole bunch of different places on a single axon or a whole bunch of different axons. So in the PNS, you myelinate with neurolemocytes, also called Schwann cells. In the CNS, you myelinate with oligodendrocytes, and that actually ends up mattering because one of them helps with regeneration if something occurs and the other one doesn't. So if you look at this figure then, where is this running through? This axon is running through where? The CNS or the PNS? It's running through the PNS because you can tell by the type of myelination it's got. One last little thing, there are little areas in between myelination where it's not myelinated, and those are called nodes or neurofibril nodes, or they used to call them nodes of Ranvier because of the dude who found them. Okay, let me tell you one little clinical connection here, which is kind of interesting. So in order to get the signal to go from here to here, you need myelination. And there is an autoimmune disorder called multiple sclerosis in which your immune system starts making a mistake. And what it does is it starts attacking the myelin sheath on your axons. You got infected with something, who knows what, you made some antibodies, and the antibodies think, unfortunately, that these neurolemocytes or Schwann cells are pathogens or invaders, and they start whooping up on them. And you lose the myelin sheath. And unfortunately, you can't get the signal to go from point A to point B if it's unmyelinated. And so what happens is you start losing sensory and motor activity or sensory and motor capacity primarily in the periphery first, where myelination happens. It happens all the way down here. So you'll lose like sensation and motor activity with multiple sclerosis, and the nervous system just starts to short circuit. Like all autoimmune diseases, we don't have a cure for it, but it can go into remission, in which you will have temporarily lost sensory or motor capacity, especially in the periphery, and then it will actually come back. So you should be able to, with the information that I've just given you, tell on this figure how to label the nodes, right? The axon, the dendrites, the myelin sheath, which is here, the axon hillock, which is that part right there, the soma or cell body, the telodendria, right? Synaptic knobs at the end of the telodendria, okay? And if you know dendrites bring information in and axons take information out, you can always look at a neuron and tell the direction of information flow, especially if it's a multipolar neuron. So the direction of information flow is that way in that neuron. Now, what happens when you get to the end is that the neuron is actually going to form a synapse with another cell. It could be another neuron, but it could be a muscle or a gland. And this one, it's one neuron forming a synapse with another neuron. This is the end of the first neuron. This is neuron 1. This is neuron 2. You can tell which direction information is going. These are dendrites. This is the axon. So what happens here is this is the site that neurotransmitters are released at the synapse. The synapse is the communication junction between a neuron and a neuron, or a neuron and another cell. So with that in mind, you should be able to look at a simplified figure like this and say, okay, where's the axon hillock? Well, the axon hillock always attaches to the cell body, so it's here. So let's call these the presynaptic and postsynaptic neurons. Let's just do this together. So this is the presynaptic neuron, because you can tell information's going that direction. This is the postsynaptic neuron. The axon hillock is here. Good. The axon, there's two. There's one axon, and here's another axon right here. The axon telodendria are here in the presynaptic. The synaptic knobs are these little guys in the presynaptic. The dendrites are here in the postsynaptic. Presynaptic, postsynaptic. The soma or cell body, I only see one of them, and it's only in the postsynaptic. The myelin sheath, I actually see two of them, pre and post, and the direction of information flow. So the cool part about understanding the anatomy of the nervous system is it really starts to tell you the physiology of the nervous system. Okay, types of synapses that exist. If it is between a neuron and a neuron, like you see here, it's called a true synapse. But there are also neuromuscular junctions in which this second cell would be a muscle, because you can tell them what to do. And neuroglandular junctions in which this second cell could be a gland, because you can tell them what to do. Okay, in the next video, I'm going to go through the glial cells, and then we're almost done.